Hello everyone and welcome to today's session. And today we'll be discussing the theory for the chapter Human Reproduction. So this is the part 3 of the lecture. Before starting this lecture, I just want to give you one information that the images, text and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So what are the objective of today's session? What we will learn in today's session? So we will understand the description and graphical representation of the steps of the gamete formation in the males. Okay, so basically we'll be talking about spermatogenesis, means sperm formation. So the male gametes, which are known as the sperms, they are produced in the testes, and there are various steps, there are various stages in the sperm formation. We'll talk about this in today's lecture. Okay, so I will show you a flow chart, okay, in which all the steps of the gamete formation in males will be shown. So we'll describe the spermatogenesis using a graphical representation in which all the steps will be listed. We will understand what is the ploidy level of the cells which are formed during spermatogenesis. Ploidy level means the number of sets of the chromosome. Clear? So we will be talking about gametogenesis today. So gametogenesis means gamete formation. So this is the broader term gametogenesis. It includes spermatogenesis and oogenesis. Spermatogenesis means sperm formation. Oogenesis means ovum formation or egg formation in females. Okay, then we'll summarize the entire lecture, and in the end we'll also discuss the DPP related to this chapter. So let's begin with this topic, gametogenesis. Now, the primary sex organs, the testes in the males and the ovaries in the females, produce gametes, that is sperms and ovum respectively, by the process called gametogenesis. So you know the primary sex organs, in males it is testes and in female it is ovaries. Now what is the function of the primary sex organs? So one of the function of the primary sex organ is to produce gametes. Another function is production of the hormones. So these are the two functions. You should remember this. Then primary sex organ in males, it is testes. There is a pair of testes in males which is present in the scrotum. Now why testes are present in the scrotum? Because in the scrotum, lower temperature is maintained. A temperature lower than the normal body temperature is maintained. Okay, so the temperature in scrotum is around 2 to 2.5 degrees Celsius less as compared to the normal body temperature, and this temperature is necessary for the spermatogenesis. Here, so the testes, it is the primary sex organ, so it will produce sperms. It will produce the male gametes. So within the testes, the sperms are produced. Now, what is the site of spermatogenesis? Where these sperms are produced? Obviously, they are produced within the testes, but where within the testes? So we have already studied, when we were studying the male reproductive system, that the testes have testicular compartments, testicular lobules, and within these lobules, seminiferous tubules are present. Okay, so within these seminiferous tubules, sperm production takes place, sperm production occurs. So answer will be the seminiferous tubules. So if someone asks you what is the site of spermatogenesis, you will write seminiferous tubules which are present within the testes. Then you can explain the structure of the seminiferous tubules. So seminiferous tubules, on the inside of the seminiferous tubules, what kind of cells or what type of cells are present? So within the seminiferous tubule, on the inner side of the seminiferous tubule, spermatogonia are present. And then Sertoli cells are present. Okay, spermatogonia. Okay, meiosis occurs in this spermatogonia to form the sperms. And Sertoli cells, these provide nourishment to these germ cells. Clear? Spermatogonia, there are kind of stem cells. Why they are a kind of stem cells? Because spermatogonia, they can self-renew themselves. They can divide mitotically and they can increase their number by mitotic division. And then these spermatogonia can also differentiate to form the primary spermatocyte, which we'll be discussing in this session. What are primary spermatocytes? What are secondary spermatocytes? Then spermatids, spermatozoa. Okay. So in females, ovaries is the primary sex organ. So ovaries also is involved in the production of the female gamete, which is the egg, and ovaries also secrete hormones such as estrogen and progesterone. Then in testes, the immature male germ cells known as spermatogonia. So what are spermatogonia? They are immature male germ cells. So there are two kinds of cells, somatic cells and germ cells. Common name for germ cells, you can say sex cells, reproductive cells. Because somatic cells, they are involved in growth. These germ cells, these reproductive cells, they are involved in the reproduction. So spermatogonia, they are immature male germ cells and they produce sperms by spermatogenesis. So meiosis occurs in spermatogonia and meiosis occurs in diploid cells. So spermatogonia, they are diploid, means they have 2n number of chromosomes. So their ploidy is 2n. Again, question can come from here. What is the ploidy of spermatogonia? How many chromosomes are present in spermatogonia in case of humans? So spermatogonia will contain how many chromosomes? Spermatogonia will contain 46 chromosomes because they are diploid. Humans, they are diploid. They are eukaryotes. So diploid number is 46. Humans have 46 chromosomes. So 2n will be equal to 46. Okay, this is the ploidy. Ploidy is the number of set of chromosomes. So 2n means it has 2 sets of chromosomes. Okay. So meiosis occurs in spermatogonia to form sperms. So sperms are produced by meiosis and meiosis is known as the reductional division. Okay. There is a reduction of chromosome number to half. Sperms, so they will be haploid. The ploidy of sperms will be haploid. Haploid means they have only one set of chromosome. Okay. Then this process is known as spermatogenesis, means sperm formation, male gamete formation. And this spermatogenesis, this begins at the puberty. So when the male attains puberty, then the sperm formation starts. Then the spermatogenesis starts within the testes. Now, why at puberty only? Why not before puberty? Because at puberty, the primary sex organs, which is the testes, it becomes active. It starts producing the hormones, which are the androgens, male hormones, testosterone, And this testosterone helps to initiate the spermatogenesis. Clear? So this spermatogenesis begins at the puberty. So at puberty, what happens? So puberty is that time of life where a lot of physiological changes take place. There is a lot of hormone production, and there is morphological changes also. Yes. So these hormones, they are controlling all these processes. Okay, so here is the diagrammatical sectional view of seminiferous tubule, which is enlarged. So you can see within the seminiferous tubule or the inside of the seminiferous tubule contains spermatogonia and the Sertoli cells. Sertoli cells provide nourishment to these germ cells, and on the outer side of the seminiferous tubule, which cells are present? On the outer side of the seminiferous tubule, in the interstitial spaces, interstitial cells are present, which are also known as the Leydig cells. And these Leydig cells, they secrete testosterone, which helps in the spermatogenesis. So you can see everything is linked here. Leydig cells, Sertoli cells, spermatogonium. So you should know the function of all of these cells and the location of these cells. Then spermatogonia, we were talking of spermatogonia, they will be diploid, means 2n, means they will have 46 chromosomes. Meiosis will occur in spermatogonia to finally form the spermatozoa, which is also known as sperm. So sperms are haploid, means they will contain 23 chromosomes. Okay, now we will understand this entire process, how primary spermatocyte, secondary spermatocyte, and spermatids are formed. Yes, from spermatogonia, primary spermatocytes are formed. From primary spermatocytes, secondary spermatocytes are formed. From secondary spermatocytes, spermatid is formed, and spermatid finally transform into the spermatozoa. Now, spermatogonia present on the inside wall of the seminiferous tubule multiply by mitotic division and increase in number. So I've told you spermatogonia is like stem cells. Okay. So these spermatogonia, they divide mitotically and they increase in the number. And some of these spermatogonia, they differentiate to form the which cells? Primary spermatocytes. Now you can see this is a diagrammatical sectional view of the seminiferous tubule. Okay, so here you can see spermatogonia are present on the inner side of the seminiferous tubule. Then you can see the Sertoli cells. These are also present on the inner side of the seminiferous tubules. Then you can see here the interstitial cells. Interstitial cells, they are also known as the Leydig cells. These are present on the outer side. They are present in the interstitial spaces. Okay, interstitial cells, also known as Leydig cells, these secrete the testosterone. These cells secrete the testosterone. Then spermatogonia, which is diploid, meiosis occurs in spermatogonia. Meiosis always occurs in the diploid cells. And due to meiosis, a haploid spermatozoa is formed, which is also known as the sperms. Then you can see this is the Sertoli cells. This provide nourishment to these germ cells, spermatogonia, and they also provide nourishment to the spermatozoa also. So we'll talk about this when we'll talk about the sperm. Okay, at the end of the spermatogenesis, we'll also talk about this. The sperm head, this is embedded in the Sertoli cells, and sperm also attains nutrition from the Sertoli cells. So that's why the nucleus of the Sertoli cells is big also. It is large because it is actively involved in essential functions during the spermatogenesis. It is providing nourishment to the germ cells. Okay, So it can provide nourishment to the germ cells because it has a large nucleus. So it has a large nucleus means it can synthesize more proteins. Okay, more nutrients. So that's why Sertoli cells have large nucleus because it is involved in essential functions during spermatogenesis, including nourishing the germ cells. Okay, this is clear. Now, each spermatogonium is diploid and it contains 46 chromosomes. Okay, this we have already studied. That some of the spermatogonia, called as the primary spermatocytes, periodically undergo meiosis. So, spermatogonium which was diploid, some of these cells of the spermatogonia, they differentiate to form primary spermatocytes. So this primary spermatocyte will also be diploid, remember. Okay, because these primary spermatocytes are formed by spermatogonia. So you can write spermatogonia, okay, they differentiate to form primary spermatocytes, which are also diploid, means they will also consist of 46 chromosomes. Then meiosis occurs in this primary spermatocytes. So here you can see this was spermatogonium. This is diploid. You can see here. These spermatogonium forms primary spermatocyte. They differentiate into primary spermatocyte. Primary spermatocyte will also be diploid, means 46 chromosome. Then meiosis occurs in primary spermatocytes to form secondary spermatocytes. So now again remember, meiosis occur in two phases, meiosis 1 and meiosis 2. Okay. After meiosis 1, 2 cells are formed, and after meiosis 2, 4 cells are formed. Or you can also say at the end of meiosis, 4 cells are formed. Okay, so keep this in mind now. Now a primary spermatocytes complete the first meiotic division, means meiosis 1. This is meiosis 1. Okay, which is reductional division. There will be reduction in the number of the chromosome. The chromosome number will be reduced to half, leading to the formation of two equal haploid cells called the secondary spermatocytes, which have only 23 chromosomes each. So this was the primary spermatocyte, which is diploid. Meiosis 1 occurs in this primary spermatocyte to form two secondary spermatocytes, and these secondary spermatocytes, it will have n number of chromosomes, means 23 number of chromosome. So from one primary spermatocytes, two secondary spermatocytes are being formed. Ploidy level is different. Primary spermatocyte was 2n, secondary spermatocyte, they are n. Now secondary spermatocytes undergo second meiotic division, means meiosis 2, to produce 4 equal haploid spermatids. Okay, so there were two secondary spermatocytes. Okay, this was primary spermatocytes. Two secondary spermatocytes will form, and then again meiosis, after meiosis 2, 4 spermatids will be formed. So these are spermatids. They will also be haploid because meiosis 2 is same as mitosis. Okay, this will be 2n, then n, and then n. Okay, so again remember, from one primary spermatocytes, two secondary spermatocytes are formed and 4 spermatids are formed. So these are 4 spermatids. Clear? This will be haploid, means it will contain 23 number of chromosomes. So now again you can see this is a diagrammatical section view of seminiferous tubule. From spermatogonia, spermatocytes, primary spermatocyte is formed, which will be diploid. Now meiosis will occur in this primary spermatocytes, in this single primary spermatocyte, to form two secondary spermatocytes. Secondary spermatocytes will be haploid. Then, okay, Then meiosis 2 will occur in secondary spermatocyte to form 4 spermatids, which will be haploid. Then spermatid will transform into spermatozoa. Okay, so this is the sperm formation. This is what is spermatogenesis. Now here is a question. What would be the number of chromosomes in the spermatid? Okay, this is a very easy question here. They're just asking you the ploidy level of the spermatids, or what is the number of chromosomes in the spermatid. So let's have a look at the answer. So in spermatid, the number of chromosomes will be 23. Means haploid. It will be haploid. It consists of only one set of chromosome. So N will be the answer. So this is because spermatids are formed after meiosis 2 during the spermatogenesis. Spermatids are formed after meiosis 2. So in secondary spermatocytes, meiosis 2 occurs, leading to the formation of 4 spermatids. So from one primary spermatocytes, 4 spermatids are formed, which are haploid. Now what happens? The spermatogonium, which is diploid, it contains 46 chromosomes. It differentiates to form primary spermatocytes, which will be again diploid. Then what happens? From primary spermatocytes, secondary spermatocytes are formed, and then spermatids are formed, and then spermatid transform into spermatozoa. Now let's have a look at this schematic representation of the spermatogenesis. So here the chromosome number per cell is given, and here the different stages of spermatocytes is given. Different kind of cells which are produced are given here. Okay. So you should remember the ploidy level, the number of chromosomes in each cell. Now spermatogenesis starts at puberty. That's why it is written at puberty. So these are the spermatogonia cells, this reddish color cells. These are increasing in the number. They are increasing their number by mitosis. Mitosis is equational division. So the cell formed by mitosis will have same number of chromosomes. So all these red color cells, they are spermatogonia, and they have 46 number of chromosomes. Okay, means 2n. Now, some of the spermatogonia, they differentiate into primary spermatocytes. So these are primary spermatocytes, but primary spermatocytes are also diploid, means they have 46 number of chromosomes. Now, in primary spermatocyte, first meiotic division occurs, leading to the formation of 2 secondary spermatocytes. So these are 2 secondary spermatocytes. These secondary spermatocytes, they will be haploid, means they will have 23 number of chromosomes. Okay. Now these secondary spermatocytes will undergo second meiotic division, or meiosis 2, to form 4 spermatids. So these are 4 spermatids, which will be again haploid. Then spermatids will transform into spermatozoa, which will be again 4, and these will be haploid, means they will have 23 number of chromosomes. So again, question can come in your exam that from one primary spermatocyte, how many sperms are produced, or how many spermatids are produced, or how many secondary spermatocytes are produced. So what you will write, from one primary spermatocyte, 2 secondary spermatocytes are produced, which will be haploid. Then from secondary spermatocytes, second meiotic division occurs in secondary spermatocyte to form 4 spermatids. So from one primary spermatocyte, 2 spermatids are produced. Or you can say from one primary spermatocyte, 4 spermatozoa or sperms are produced. Okay, so primary spermatocytes, they undergo meiosis 1 to form secondary spermatocytes, which will be haploid. Then secondary spermatocytes undergo meiosis 2, which is also known as it is like mitosis. Meiosis 2 is like mitosis. Means it is kind of equational division. And this will give rise to spermatids, which will be haploid, means they will have 23 number of chromosomes. Okay, we have already discussed this a graphical representation of spermatogenesis. Okay. So they are showing you three things here: chromosome number per cell, and then they have shown you spermatogenesis using these cells, and they have told you what kind of cells are produced during the spermatogenesis. Okay, this is clear. Now there are two terms, spermiogenesis and spermiation. Let's talk about this. First, let's talk about the spermiogenesis. So it is a process where spermatid transform into mature spermatozoa. So when these spermatids they transform into mature spermatozoa, this is known as spermiogenesis. Okay. And what is spermiation? So spermiation is the release of mature spermatozoa into the ducts of the seminiferous tubule, into the lumen of the seminiferous tubule. This is known as spermiation. So there are two terms, spermiogenesis and spermiation. What is spermiogenesis? The transformation of the spermatids into spermatozoa. And then what is spermiation? Spermiation is the release of the spermatozoa or release of the mature, you can say sperm, into the lumen of the seminiferous tubules. This is known as spermiation. Okay. Now from the seminiferous tubule, these sperms will travel through the accessory ducts. Okay. So after seminiferous tubule, the sperms will enter what? They will enter into retitestes, then vas efferentia, then epididymis, then vas deferens. Then finally, vas deferens is connected to the duct coming from the seminal vesicles. Okay, it forms ejaculated duct, and through the ejaculated duct, the sperms are, you can say, transported out of the body through the urethra. Okay, this is clear now. This was spermatogenesis, means sperm formation or male gamete formation, which occurs in the seminiferous tubule, which is present within the testes. So spermatid contain half the number of chromosomes compared to somatic cells. So as I have told you, there are two kinds of cells, somatic cells and the germ cells. So somatic cells, they are diploid. They have 2n number of chromosomes. They will have 46 chromosomes. These somatic cells, they are involved in growth. Okay. Germ cells, they are involved in reproduction. So spermatids, which are haploid, they will contain 23 chromosomes. So spermatids contain half the number of chromosomes compared to the somatic cells. Okay. Each chromosome in a spermatid is single. Each chromosome in a spermatid has a single chromatid. So what does this mean? Okay. So in diploid condition, what happens? Two sets of chromosomes are present. Means the chromosome have two chromatids. So this is a chromosome with two chromatids. This condition is in 2n, diploid condition. In haploid condition means n, what happens? A chromosome has only one chromatid. So this is a chromosome with only one chromatid. Okay, this is in the 2n condition. Each chromosome will have two chromatids, and these two are known as the sister chromatid. Okay, here you can see. Clear? So each chromosome in a spermatid is each chromosome in spermatid has single chromatid only. Now these spermatids, they mature into functional spermatozoa, maintaining the haploid number. This process is known as spermiogenesis. Now, why the gametes are haploid? Why not diploid? Why the gametes are not diploid? Why they are haploid? And why meiosis is occurring during the gamete formation? So you can see these gametes, they are haploid. Okay. So what happens after the gametes are formed? After the gametes are formed, the male and the female gamete, they fuse with each other. Fusion of the gametes, it is known as fertilization. So after fertilization, again, the diploid structure, zygote, is formed. So the chromosome number is maintained. Okay. So this is the significance of meiosis also. Maintenance of the chromosome number. Okay. So this is the male gamete. One is the male gamete. Another one is the female gamete. Okay. Male gamete and the female gamete. Both have n number of chromosomes. In case of humans, both have 23 chromosomes. They will fuse together to form 46 chromosome, means diploid condition. So a zygote is formed. This zygote will have 46 chromosomes. In case of humans, it will be diploid. So you can see the chromosome number is maintained. Okay, that's why gametes, they are haploid. That's why meiosis occurs, to maintain the chromosome number. Okay, and variations also occur. So during meiosis, what happens? In the Pachytene stage, crossing over and recombination occurs. Okay. In recombination, what happens? There is exchange of genetic material. And this is a source of variation. Okay, so you should remember all of this. Gametes are formed due to meiosis. In meiosis, recombination and crossing over occurs. This is a source of variation. Meiosis maintains the chromosome number. And the gametes are haploid again, because when the gametes will fuse, they will restore the chromosome number of that species. If the gametes are diploid, then what will happen? Okay, if they are diploid, if they are 2n and 2n, so they will fuse to form 4n. Okay, then they will again produce gametes 4n and 4n. So zygote will be 8n. So you can say the chromosome number of the species is not maintained here. That's why meiosis is very important. Now, this haploid number of the gamete is essential for normal fertilization, as it combines with the egg cell's 23 chromosome to form a diploid zygote. Okay, this we have already discussed. The chromosome number is maintained now in the zygote. Here. Now let us examine the structure of the sperms. Okay. So sperm is microscopic structure composed of head, neck, middle piece, and a tail. You should remember the parts of the sperm. First one is the head, then neck, then middle piece, and the tail. Now we'll understand all these parts. Okay, the first one, a plasma membrane envelops the whole body of the sperm, first of all. So a plasma membrane is present, and it is around the whole body of the sperm. Okay. Sperm head, let's talk about the sperm head. It contains an elongated haploid nucleus, and the anterior portion of the head is covered by a cap-like structure known as the acrosome. Okay. Clear? So the sperm head, it consists of the nucleus. And within the nucleus, the chromosomes are present. The genetic material is present. Clear? And you can see the anterior portion of the head, it is covered by a cap-like structure known as acrosome. So this acrosome consists of enzymes. Acrosome enzymes. Okay, and these enzymes help in the breakdown of the egg cell membrane. Okay, it digests you can say the membrane of the egg, and it helps in the fertilization. Okay, so this is known as acrosome reaction. So this acrosome reaction is very important for the fertilization of the egg. And this acrosome, which is a cap-like structure, in this acrosome, the enzymes are present which digest the wall of the egg. Enzymes are like hyaluronidase. Okay. Hyaluronidase. This is the enzyme which is present in the acrosome. Okay, so this was about the head of the sperm. Now, acrosome is filled with enzymes like hyaluronidase and acrosine. So these are the enzymes present in the acrosome. And these enzymes help with the fertilization by assisting the sperm in penetrating the ovum's protective layer. So there are some layers which are present around the ovum. like zona pellucida, corona radiata. We'll talk about these layers in our next sessions. So these enzymes, they help in the breakdown of these protective layers and help in the fertilization process. Okay. After fertilization, what is formed? A zygote is formed, which is diploid, means it will have 46 number of chromosomes. Okay. The zygote will then form the embryo. Okay, which will again contain 46 chromosomes now. So from a zygote, which consists of 46 chromosomes, a normal child will develop. Okay, a normal child contains 46 chromosomes. Clear? Now I've told you if gametes are not haploid, if they are diploid, if no meiosis occurs in the formation of gametes, then what will happen? Usually the gametes are haploid, they are N and N. So they form a diploid structure 2N. Okay. So from this, a normal child will develop. If the gametes are 2N and 2N, then the zygote will be 4N. So this is not normal. Okay, a normal child will not develop from this 4N zygote. So this is the structure of the sperm. So you can see the head of the sperm here. It consists of the nucleus containing the chromosomal material. This nucleus fuses with the nucleus of the egg and leads to the formation of the zygote. Okay, so this is again a source of variation. The genetic material of the male and the female gamete, they are fusing together. Okay. Now here you can see on the anterior part, the acrosome is present. Acrosome consists of the enzymes like hyaluronidase and acrosin. Then plasma membrane surrounds the entire surface of the sperm. Then below the head, neck is present. Then middle piece is present. What is special about middle piece is middle piece contains numerous mitochondria. And this mitochondria, they are involved in ATP generation. So they are the source of energy for swimming. Because this sperm, this also have a tail. This tail helps in swimming. And for swimming, energy is required. And this energy is provided by the mitochondria. So that's why it is written mitochondria. This is the energy source for swimming. The numerous mitochondria present in the middle piece. Okay, sperms, they are motile. The egg cell, the ovum, that is non-motile and that is large also. Sperm, they don't contain much cytoplasm. The egg cell contains more cytoplasm. It is of larger size because the ovum have to form the embryo later on. Now the middle piece possesses numerous mitochondria which produce energy for the movement of the tail. So this movement requires energy. So how these sperms travel? So after the insemination, means after the transfer of the you can say sperms into the female genital tract. This is known as insemination. The sperm travels from the vagina to the cervix, okay, and then finally to the ampulla region of the fallopian tube, and in the ampulla fertilization occurs. Okay, so for the sperm motility, energy is required. This energy is provided by the mitochondria. If the sperm can't swim, then there will be no fertilization because sperm have to travel and it have to reach the ampulla region of the fallopian tube where egg should be present. Then only the fertilization will occur. A human male ejaculates about 200 to 300 million sperms during a coitus. Coitus means sexual intercourse. Of which, for normal fertility, at least 60% of the sperm must have normal shape and size, and at least 40% of them must show vigorous motility. Okay, so this is the condition, okay, for normal fertility. The 60% of the sperm should have normal shape and size, and 40% should be, you can say, show vigorous motility. Now, sperms released from the seminiferous tubules, they are transported by the accessory ducts So what were the accessory ducts? Retitestes. Okay, then vas efferentia, epididymis, vas deferens. So these were the accessory ducts. So when the sperms are released from the seminiferous tubule, they will enter into the retitestes. From retitestes to vas efferentia. So vas efferentia will carry the sperms to epididymis. In epididymis, what happens? There is a storage and maturation of the sperm. So storage and maturation of sperms occur in epididymis. Then from epididymis, the sperms, they are transported by the vas deferens, and the vas deferens is connected to the duct of the seminal vesicles, which forms the ejaculatory duct, and from ejaculatory duct, the sperms are transported to outside of the body through the urethra. Again, we have discussed about the structure of the sperm. These are the different parts of the sperm: the head, neck, middle piece, and the tail. Now, secretion of the epididymis, vas deferens, seminal vesicles, and prostate are essential for maturation and motility of the sperms. Okay, epididymis, vas deferens, these are the accessory ducts. Seminal vesicle, prostate, these are the accessory glands. Now, male accessory glands. What are the main accessory glands? So, there are three male accessory glands: prostate gland, seminal vesicles, and bulbourethral gland. So these glands produce secretions that contribute to seminal fluid, which provide nutrients, transport medium, and other substances to nourish and protect the sperms. So secretion of these accessory glands are very important. And secretion of these glands is known as seminal fluid or seminal plasma, which is the major component of the semen. Okay. Now, what is the role of seminal vesicles? So seminal vesicles, they secrete seminal plasma. Okay, their secretions consist of, you can write, fructose and prostaglandins. So secretion of seminal vesicle contains fructose and prostaglandins, and this fructose, you can say, provide nourishment to the sperm. Then the secretion of the prostate gland. The prostate gland secretes a milky alkaline fluid, which is again a major component of the semen. This bulbourethral gland, this lubricates the penis or lubricates the urethra and also neutralizes the acidity, which is caused due to urination. And the neutralization of acidity is very important for the viability of the sperms. Seminal plasma along with the sperms constitute the semen. So secretion of all these glands, okay, all these accessory glands, is known as seminal plasma, and this is the major constituent of the semen. Okay, you should know the individual role of all of these accessory glands. Now, the function of male accessory ducts and glands are maintained by the testicular hormones known as the androgens, which are male hormones such as testosterone. Now, let's summarize this entire lecture. Spermatogenesis results in the formation of the sperms that are transported by the male sex accessory ducts. A normal human sperm is composed of head, neck, middle piece, and tail. So, you should know the structure of the sperm as well. You should know why the mitochondria are present in the middle piece and how the energy is provided for the motility of the sperm. Okay, you should know about the ploidy level of all the cells which are formed during the spermatogenesis. Okay, and then from one spermatocyte, one primary spermatocyte, how many spermatids or sperms are produced. Clear? So this is the end of the theory. Okay, we have discussed about the spermatogenesis. Now let's discuss some DPP related to this theory. Okay, let's discuss the DPP now. Now, first question: spermatogenesis starts at the age of puberty due to significant increase in the secretion of. Spermatogenesis means sperm formation. It starts at the puberty in case of males. Okay. So for spermatogenesis, which hormone is required? Or there is a significant increase of what? So these are the options: somatostatin from hypothalamus, gonadotrophic releasing hormone from hypothalamus, gonadotropin releasing hormone from anterior pituitary, and sex corticoids from the adrenal gland. So what will be the correct answer? With the correct answer here will be gonadotrophic releasing hormone from the hypothalamus, means option number B is the correct answer. So what happens? Hypothalamus it releases this hormone, gonadotrophic releasing hormone. This hormone, it stimulates the anterior pituitary, okay, to release gonadotrophins, which are known as follicle stimulating hormone and Leydig hormone and luteinizing hormone. Okay, so FSH and LH, they are released by the anterior pituitary because it is being stimulated by the gonadotrophic releasing hormone from the hypothalamus. Now, this luteinizing hormone, it further stimulates the Leydig cells to secrete testosterone involved in spermatogenesis, and follicle stimulating hormone. This hormone stimulates the Sertoli cells, and the secretion of Sertoli cells are also important to initiate the spermatogenesis. So answer will be option number B here. Now next, which hormone is responsible for initiating spermatogenesis at the puberty? Again, this is a related question here. They're asking about initiation of the spermatogenesis. They are directly asking which hormone. Again, the options are follicle stimulating hormone, luteinizing hormone, gonadotrophic releasing hormone, and testosterone. Okay, the correct answer here will be option number C and not D here, testosterone. Testosterone, yes, it is involved in the spermatogenesis. But here we will write gonadotrophic releasing hormone. This is the most appropriate answer here. Again, what happens? We have already discussed gonadotrophic releasing hormone released from the hypothalamus. This will stimulate the anterior pituitary to release FSH and luteinizing hormone. Luteinizing hormone will stimulate Leydig cells to secrete testosterone, and follicle stimulating hormone will stimulate Sertoli cells, and secretion of Sertoli cells, they are very important to initiate this spermatogenesis. Okay, so you can see the four options, they are very close to each other. All these hormones, they are involved in the spermatogenesis. Clear? Answer will be option number C here. Then how many chromosomes are present in each spermatid formed during spermatogenesis? So they are asking you about the ploidy level of the spermatid, the number of chromosome in these spermatids. We know 4 spermatids are formed from one primary spermatocyte. Okay, so how many chromosomes are present in spermatid? 23, 46, 92, or 12? So, humans, they consist of 46 chromosomes. So they have diploid cell. These diploid cell consist of 46 chromosomes. So keeping this in mind, you have to answer this question. The correct answer here will be option number A, 23 chromosomes. So spermatid, they are haploid. The haploid cell in case of human, they consist of 23. They are produced by meiosis. Okay, meiosis reduces the number of chromosome to half. So diploid cells, they have 46 chromosomes. The haploid cell they will contain 23 chromosomes. Now next question, what is the role of acrosome in a sperm? So acrosome, this is present at the anterior end of the sperm head. Okay, anterior end, you can say, or the upper end of the sperm head, you can say. So what is the role of this acrosome? What is the function of the acrosome? Options are: provide energy for motility, contains enzyme for fertilization. Third is protects the nucleus. Fourth is facilitates the tail movement. The correct answer here will be they contain enzymes for the fertilization. Option number B is the correct answer. So acrosomes, they contain enzymes which helps in the breakdown of the egg membranes and help in the fertilization. So we'll discuss about the acrosome reaction in our upcoming sessions also. Okay, so in acrosome reactions, there are a lot of steps involved. The acrosome membrane, it binds with the plasma membrane of the sperms. Then these, you can say acrosome proteins, they are expressed. And these acrosome proteins, then they recognize the proteins present on the zona pellucida, specifically ZP3. Okay. Clear? So these are some of the steps in the acrosome reaction. We'll talk about this in our upcoming sessions. Now, next question: which structure in the sperm contains mitochondria? Okay, which is necessary for motility. So mitochondria present in which part of the sperm? Head, neck, middle piece, or tail? Okay, the correct answer here will be mitochondria. They are present in the middle piece, means option number C is the correct answer. The numerous mitochondria present in the middle piece. These mitochondria are the source of energy. Clear? Head contains of the nucleus and the anterior portion of the head consists of acrosome. Clear? Tail is there for the motility. Clear? It provides motility. So middle piece is the correct answer here. So we have discussed all the DPP also. Clear? So all our answers are correct. So now this is the end of this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session.